Cancer Drug and Research Overview. This is the first and foremost needy for your career. So I hope this session will be a good platform for our students. My dear students, enthusiastic students, kindly utilize this great opportunity and explore your knowledge. Once again, I welcome you all. All the very best, my dear students. Thank you, madam. Thank you, madam. Madam, Preetha, madam, Dr.
Yes, is
Preetha,
that my
madam.
cue? Yes, yes, I'm
Yeah.
here.
Please, madam, you can proceed, madam.
All right,
Continue,
thank you. Thank
please.
you for the very kind words. First of all, I just wanted to say something. Yeah, thanks to Shiva. Shiva was the one. Shiva's a long-term friend of mine. We have been friends since I was in masters, like when I was very young. Can you hear me? Okay.
Yeah, yeah.
Yeah, and I think Shiva is online too. Hi, Shiva. Anyway, so quickly, I'm not a doctor, so there's a disclaimer there. I don't have a PhD. I have a master's degree in biotechnology. I just wanted to put it out there before I start. So give me one sec. Let me start my slides.
Anbu sir,
சார் சொல்லுங்க சார்.
என்னாச்சு?
இல்ல மேடம் ஸ்லைட் ஷேர் பண்ணிட்டு இருக்காங்க சார்.
சரி, சரி, சரி. ஓகே. சிவா சார் இருக்காரா சார்? சிவா லைன்ல இருக்காரா? சிவா அண்ணன்மார்.
இல்ல சார், இல்ல சார்.
ஓகே, ஓகே. அவருக்கு இன்விடேஷன் ஷேர் பண்ணியிருந்தேன், அதான் கேட்டேன்.
ஓகே சார், ஓகே சார். சார் மேடம் இன்னும் 2 மினிட்ஸ் ஜாயின் பண்ணிடுவாங்க சார்.
ஓகே சார், ஓகே சார். அட்டெண்டீஸ் ரொம்ப கம்மியா இருக்காங்களே சார். அன்பு சார்.
எஸ் சார், எஸ் சார். 125 128 தான் இருக்காரு சார்.
131.
ஆமா சார்.
புரியல.
சார்
அதெல்லாம்
சொல்லுங்க சார் வணக்கம் சொல்லுங்க சார்.
ஒன்றும் இல்லை சார், ஒன்றும் இல்லை சார்.
ஓகே சார்.
மேடம் ஜாயின் பண்ணிட்டாங்களா இல்லை என்ன?
I'm here.
2 minutes, madam. I'm downloading the PowerPoint.
Sure.
Okay.
Yeah, I'm sorry about the technical glitch. I think I don't know what's going on. I think it's my organization is trying to block off the content sharing thing. But anyway, so I have sent my slides to Anbu, and he will put the slides on the screen, and I'll just ask him to navigate the slides. It shouldn't be a problem.
Okay, madam. 2 minutes. 2 minutes.
Sure. Take your time.
Two minutes, madam. I have downloaded. I will start up.
Okay, no worries. Yeah, it is intimidating talking to 150 people. But can you hear me okay?
Yes.
Okay.
Madam, I can't open your file. Can you able to reset it again?
Oh, okay. Do you know what's going on?
No, the file is not opening.
Huh. That is weird. Hold on. That is strange. The file opens here. Okay. Let me. I've sent to you again.
Okay.
Let me know. Let me try something else too on the side.
Definitely.
Let's try this. If the one that I just sent you does not work also, try the third email that I just sent to you.
But recheck it. Can
Yeah,
able
yeah.
to see the screen?
Okay, beautiful. Okay. It's good to see something on the screen. All right. Okay, do you want to do the slideshow?
Yes, madam.
Okay. Are you all working from home or?
We are working in the home only.
Okay.
Yes, madam.
No, I was just curious.
So can you? It was in the full screen, madam.
No, no. I was just going to ask you if you were put the slideshow on.
Yeah, I have put the slideshow only, but
No, this. Yeah. Okay, so you want me to just go ahead start or?
yes, madam, yeah, you can proceed with that, madam.
Okay, or do you want to blow it up? Like what I mean is, if you go to the right bottom, you see three little icons, like right bottom of the screen,
No,
where
madam,
it
I know
says.
that, but there's
Okay.
a clear example.
Okay. So I'll just, okay, because I had a couple of very small animations that might help, but that's fine. That's fine. Okay, I'll just go ahead, start. I'm really sorry about all the technical glitches that happened, and I'm really sorry about the delay. I think it's because of the organizational thing. My organization, that because I work in a hospital, they are crazy about paranoid about privacy, and that's, I'm guessing, is the reason why I could not share my content. So yeah, but I'm glad that you guys, everybody was patient with me while we worked everything out. So anyway, let's start to start. So I'm Preeti Ravindranathan. I have a master's. I do not have a doctor degree. I don't have a PhD. But I've been in research for a long time. I started in cancer research. I was in cancer research for 13, 14 years until recently, like 3, 4 months back, I moved to cardiovascular research, which is a lot of new learning for me. It's fun too. So let me get started. So today, what I thought of talking to you was about targeting cancer. Um, I was specifically going to talk about. You can move on to the second slide.
Yes, madam. I think there is an issue.
Oh, okay.
We'll start to reset again.
Uh, all right. So yeah, like I was saying, because it's a huge topic, cancer research and what's going on in the cancer research field is, it's vast and it's impossible to cover in one session. So I thought I'll narrow it down to just two things. First, of course, what is cancer? The second is I'll touch on two things that is very hot right now. One is immunotherapy to combat cancer, and what's going on right now in preclinical and clinical research. And the second thing, I'll also talk about how clinicians and scientists are trying to overcome drug resistance in cancer. Next slide. All right. So yeah, like I was saying, the first step in fighting something is to know what your enemy is. Only if you know something can you fight it. So next slide. So so I decided to give you a quick overview of what cancer is. So what cancer is, if someone asks you what cancer is, you might say it's uncontrolled proliferation of cells. This cells growing like crazy without any regulation, right? So that is primarily what people were thinking earlier, like for many, many years. That's a reductionist view that I have on the left panel. That's showing there's a lot of cancer cells growing uncontrollably and just exploding. But then, as research happened and as people started knowing a little bit of things in different areas within cancer research, they understood that it's not just as simplistic as cells proliferating. There's a lot more to cancer. So it's like it's it's a it's a whole system orchestrating in to do something to become cancer or tumorigenesis. So that's what I'm trying to show in the second, the right panel. So yeah, you can move to the third, next slide. So yeah, so again, I'm just expanding on the same thing, that in a cancer, there's a lot of things going on. A lot of different cells are trying to orchestrate the whole event and trying to progress the cancer. So I can barely see, but if I remember right, it's not just the cancer cells. You have the fibroblasts, and the PC cells, the cancer stem cells, and other things that are working together. So the other thing is, so once this cancer is established, it cannot stay in one single place. It has to grow and then move to another place. So it needs to metastasize. It needs to invade into another place to find a new home, and that's how cancer spreads. That's how it flourishes. And for all this to happen, there needs that in the bottom panel. That's what I'm trying to show. I'm trying to show that um, not just the cancer. There are things that is happening at the new site uh, that is making the cancer deposit there and then flourish there. So again, I'm just trying to reiterate that there is a lot of events happening, and there are not just the cancer cells, but there are so many other things that are helping it progress. Next. All right. So saying all this, so this is from 2000. This is a famous picture. This is a famous argument that was put forward in 2000 by Hanahan and Weinberg. So what they are trying to say is, what they put forward was that there are at least 6 characteristics of cancer that must happen for it to flourish, for it to survive. So if you look at it, I'll start with the green one, the proliferation signal, right? So the cancer, of course, it needs to grow uncontrollably. That's the main hallmark of cancer. The second is the brown. I'll go clockwise. So the second is it needs to evade the growth suppressors. Give me 1 sec. Let me pull up my PowerPoint because I can barely see. Give me 1 sec. All right. Okay, that's much better for me. All right. So the second characteristic feature of a cancer is that it needs to evade growth suppressors, right? So whenever there's something growing out of control, other cells will come and try to suppress it, give it suppressing signals, but the cancer will have to bypass all those to be able to progress. And the third one is, of course, it needs to activate the invasive and metastasis, which is what I was just talking to you about. It needs to get out of the old home and then make new homes out of new places, new locations in the body. And of course, the fourth one is replicative immortality. So it just needs to keep replicating again and again, so without any stop. So if you know this, your normal cells, after a few number of replications, it stops dividing. That's how the body controls the number of the growth of any cancer cells. But then cancer cells, to become cancerous or to become genic, it needs to be able to infinitely replicate. And then the next one is inducing angiogenesis. That's another characteristic, because as the cancer grows, it becomes really lumpy, and to be able, it needs, of course, nutrients, right? So it needs blood flow to it, but then it does not have enough blood flowing to it, so it needs to create its own blood flow. So what usually cancers do is, in the initial stages of tumorigenesis or pathogenesis, they will create new blood vessels to themselves. So that is another hallmark feature of cancers. And lastly, it needs to resist cell death, which means that, again, it's very similar to evading growth suppressors. So apoptosis is programmed cell death. So this is something that happens to all cancer cells, any normal cells. Give me 1 sec. My dogs are here. Yes, I'm sorry. I'm back. The dogs were here. Right. So in short, these are all the 6 different things that must happen for a cancer to successfully survive. Move to the next slide, please. Okay. So what I showed you in the previous slide is an older view of cancer. But then, as we have more research into the whole thing, we see that there are newer things that should happen in cancer for it to be successful. So those things, and again, avoiding immune destruction, which means that, I mean, whenever there's a cancer growing in the body, your immune system will get activated. It understands that there's something wrong with the body, and that therefore it comes to fight off the cancer. So for the cancer to become successful, it needs to fight this off. So that is another characteristic feature. This is very new, and this is what we'll be talking about today. Kind of. So I will be touching a lot on this area and how we are trying to hack into this trait of cancer. And apart from that, deregulating cellular energetics, that's another area. I don't want to touch upon that, but basically what it is trying to say is, because cancer is growing so fast, it needs to have its own way of having of how do you say this, energy mechanism. So what it does is it reprograms itself so it can I'm sorry, it can I'm trying to so it can get a lot more, it can use energy efficiently, or it just reprograms itself to get more energy from whatever energy source it's getting. Yeah. Next slide, please. And boom. Okay. Okay. So I was showing you all these things. Again, reiterating, all these different characteristics must happen for the cancer cell for the cancer to be successful. But it does not necessarily mean that everything has to happen at the same time, or or there are different times. Okay, let me back up. What I'm trying to say is, if you look at the slide, the left side of the slide and the bottommost, you see that that is the shortest path for a cancer to develop. So, which means that there are just five things happening, five different mutations happening, and that is giving the cancer the characteristics that it needs to become successful. On the other hand, if you look at the topmost pathway, there are like more than five or six, one, two, three, four, five, six, seven, eight. Yeah, there are 8 items. There are 8 mutations or genetic lesions that are happening, which is conferring the cancer a little bit of the characteristic at one time, to ultimately have all the different 8 characteristic to make it successful as a cancer. Am I going too fast? I know this might be a little confusing, but just bear with me. This is just some basics that might help you understand where I'm going with next. Next. We can skip this one. Next. All right. So what are we going to do with all this knowledge that we have? So we know how cancer functions. We know what all the essential things that should happen in a cancer for it to become successful. So what do we do with all this knowledge? Of course, we strategize based on all these things. So in this slide, what they're trying to show is, so we are trying to attack every different aspect of the cancer. So if you look at it, so in the outermost circle, right, these are the different strategies of how scientists and clinicians are trying to attack cancer. So they take, for example, I'm trying to pick one from, okay, let's take the green one, sustaining proliferative signaling, right? So that is one hallmark of cancer. So what we are trying to do, by we, I mean the cancer researchers and scientists, what we are trying to do is find inhibitors of those proliferative signals for cancer. So one example is EGFR inhibitors. EGFR is one proliferation signal. So that is very important for cancer. Cancer is almost addicted to it. So what we are trying to do is inhibit EGFR. So that's one way of attacking cancer. So this whole picture just shows you all the different ways people
And your voice is not audible. Sorry for the interruption.
Sir?
Sir, vanakkam sir. Solunga sir.
Hello.
Yes, yes, yes. Yes, madam.
I'm so I'm
Please
sorry
continue.
about this. I don't know
No,
what's
no,
going on.
no. No issue, no issue.
Uh-huh.
No
Yeah,
issue.
third time is a charm. So hopefully this time it will work. This time I connected from my phone, so
Yeah,
hopefully
yeah.
it should be okay.
No issue.
Yeah.
Please, madam. Just continue.
Okay. I was just getting to the fun part of my whole talk. So anyway, move to the next slide, please. Yeah. So this is the actual fun part of the talk. So how do we attack the cancer, the spread of cancer? Next. So traditionally, how it is done is, of course, there are three prongs to how cancer is treated. First is surgery, of course. They remove the mass of the cancer. The second is radiation, where they irradiate the tumor so it'll shrink and then stop growing. The third is chemo drugs, of course, chemotherapy, which most of us are familiar with. But then I'm not going to touch upon any of these. I'm going to talk about the hottest in today's therapy research, and there's a lot also growing in standard of care also, which is immunotherapy. Next. So the first immunotherapy so I'll just talk about a few things here within immunotherapy. Immunotherapy is basically hacking your immune system to work for you and fight the cancer off. So the first one is the ADCC, or antibody-dependent cell-mediated cytotoxicity. So what is done here is you take the cancer cell, and of course you have your immune system, the NK cells from the immune system. But unfortunately, for some reason, the cancer cell is hidden from the immune system. So the immune system is not able to see or attack it. So what we would do is send give an antibodies. It's like a mediating thing. So it's what inject these antibodies um, into the cancer body, the patient, right? So this antibody will bind both the NK, your immune system, and also the cancer system, and therefore make the cancer cell get unmasked, and therefore the NK cells will take care of it. So this is just one example. So there are several of these FDA-approved drugs which is in the market right now, and people are actually, there are, of course, clinical trials, and of course, there are also many standard of care treatments that is going in using this method. Rituximab is a very popular one that we were working on recently in my department. And the development here, what they're trying to do is, of course, we have good drugs like rituximab and lotuzumab, but then what they're trying to do is create more antibodies which have better affinity to bind both NK cells and cancer cells. Next. So this is really hot, and especially I don't know if you were familiar. In 2018, this was what got the Nobel Prize in Medicine, Dr. Jim Allison, and I forget the other Japanese scientist's name. But so this is really hot now. This is where everybody most people are working on right now. So what this is again, the cancer cell is trying to evade immune system by being hidden from it. So again, if you look at the left side, you see so this is one way how the cancer cells do this. So I'm not sure how familiar you are with T cells and how T cells act. You don't need to know too much, but then basically the idea is that whenever there's a T cell, it usually will see the tumor cell and try to attack it. But the tumor cell, what it does sometimes sneakily is that it'll produce PD-L1 or some other inhibitor, some other antibody, and this PD-L1 will bind to PD-1 on the T cell. And this makes it pretend like it's a friend, it's nothing serious, and that's how it evades being attacked by the T cell. So this is very sneaky of the tumor cells. So what the scientists and the clinicians are doing right now is hacking this mechanism. And what they do is bring something in between PD-L1 and PD-1, therefore something like an antibody, something that binds between these two and does not allow them to bind together. And therefore, the T cell remains active, and therefore it's able to prompt immune system, immune response, and therefore attack the tumor cells. So again, Pembro and Nivo are very, very popular and very promising drugs right now. And we, again, were doing some trials with these. Next. So again, this is just an example of other checkpoint inhibitors. Yes. So like I was saying, James P. Allison, he and Tasuku Honjo, he's a Japanese scientist that I just mentioned about. So these two scientists, they got 2018's Nobel Prize for Medicine for their work on this. So one of them, Allison Allison's work is on anti-CTLA, which is also a checkpoint inhibitor. Very similar mechanism. And Tasuku Honjo's is anti-PDL1, which is what we just talked about. So now what people are trying to do is combine these two. These are like two different checkpoint inhibitors, but they're trying to combine these two slightly different mechanisms to see how that works. And apparently, this is really promising. And if you have the time, I would really urge you to go and look at the different um, trials and how promising the combination of these two trials is. Next. All right, the cancer vaccines. So this is another topic that's kind of fun. So what they do here is this is exploiting certain peptides that is very specific to cancer. So these are called neoantigens. If you look at the picture, these are shown in small green little structures there. So these can be synthesized. These we know how they look and everything. So they are synthesized in the lab or in petri dishes, whatever, or chemically, and they are put into dendritic cells, which are immune cells, of course. This dendritic cells, I don't know if you again how much immunology you have had, but then dendritic cells are something that'll take something in, chew it up, and then present it to T cells in a fashion that it understands. So that's what dendritic cells do. So anyway, so it is doing all the processing for it and then provides it to the T cell. And now the T cell knows that, okay, this peptide is something that I need to be aware of. So when this T cell goes back to into the body, if you look at it, the T cell is now able to recognize all those dirty peptides in the cancer, and therefore it's able to attack the cancer. So this is another promising thing that's happening right now. Next. All right. So I told you that we have the neoantigens, and we make the dendritic cells process and give it to the T cells. But then this can be done in so many different ways. How do we make the dendritic cells process this? So one example is just take the person's, because dendritic cells are, of course, immune cells, right? So you just take the person's monocytes, which contain the dendritic cells, and then isolate the dendritic cells from him, combine them with the neoantigens in a petri dish, in a lab setting. And the dendritic cell, even outside the body, does its magic. It does the processing, and then you combine everything, you put it back into the cancer patient, and it works magic. So that's one way of doing it. The other way is, of course, just take the synthetic peptides, the neoantigens, and then without taking it out of the body, the monocytes out of the body, just directly put it inside the person's body, and the monocytes inside the person's body will do the same magic. It will process it and then present it to the T cells. And that way, the T cells are able to counter the spread of cancer. So next slide. So that's one way of doing it. And now there's a fancier version of the whole thing. We have DNA and RNA vaccines. So it's the same thing. It's just a different way of presenting those neoantigens, right? So here, the DNA vaccines, it's just that the peptides, the neopeptides, instead of putting them as peptides, we give them as DNA sequences, which can create those peptides. So those are put inside plasmids, and then those are given into the cells, and those peptides, those plasmids will go in and produce those peptides, and then dendritic cells will recognize them, process them, and that happens. The same thing happens with RNA vaccines, very similar concepts, except instead of DNA, we are giving it as RNA. Next. Oh, did you already move to the next? Okay. So this is a further advancement. This is where a lot of activity and a lot of fun things going on right now, because this is where you might want to go back one step with the slides. Right, thank you. So this is where cancer vaccine is merging with computational biology. So we have a lot of knowledge we know a lot about genomes. We know a lot about DNA sequencing and RNA sequencing has advanced so much that you can actually do sequencing a lot. So now that we have a reference genome, and then of course you can do sequencing fast, so you can take the cancer patient and then sequence his tumor DNA and sequence his normal DNA and identify the DNA which is kind of different in cancer DNA. So that way you know what is different about cancer in that person. This is personalized medicine. So you can do that, and then you take that, you do some computational modeling, see which one will bind to the MHCs, again computationally, and then do the real test, put it inside tumor cells and then into MHCs, do the same thing, and then back again into the body. So this is again, this might look like out of, I mean, this might look like very strange, very next generation thing, but this is already in the works. People are already working on it, and there are already many early phase clinical trials in this area. So this is kind of cool. Next, CAR T cell therapy. So this is, I don't know, again, if you've heard about this, but CAR T cell therapy is, again, very hot right now. When this started a couple of years back, we were the first ones, a few years back, we were the first ones to do the trials in the whole of the United States in our institute. But then now it's many people are doing it. This has become very, very common. So this is, again, very similar, but slightly different. So what happens is, again, you take the T cells out of the person, the cancer patient, and then insert the CAR gene into the T cells. Again, this is like educating the T cells. So the CAR gene inserted T cell is called the CAR T. And then these are grown in a petri dish inside the lab and then multiplied. So the population grows, and then that is given back into the patient, and the same thing happens. Now the T cells are alerted to cancer, and therefore they're able to fight cancer. So all these are about, it's all the same mechanism, the same kind of theme, but then it's just very different, small nuances to each different therapy. Next. Okay, so this one, CAR T cell therapy. I just wanted to show this to show how advancements happening within the CAR T cell therapy, right? So the first generation of CAR T cell therapy just had an antigen recognition domain and a signal which will make it produce within T cells. But then now with better research, and then now we have other generations where you have more signals inserted into this, and therefore it's just giving it better ways to gives it better qualities. And yeah, so what I'm trying to say is CAR T is really evolving. There's a lot going on here. Next. Okay, so that is about the immunotherapy. So I talked about a few exciting things that are going on right now. The second thing that people are struggling to tackle, and there's a lot of activity, is in overcoming cancer drug research. Because I don't know if you have seen any cancer, if you know anyone with cancer or an acquaintance with cancer, you probably know initially when the treatment is given, they will respond to it. The tumor shrinks and everything. So good things happen. And then all of a sudden, it just goes out of control. The tumor starts growing again. It comes back with vengeance. And then when you give drugs to the tumor, nothing happens. It just continues growing, and then it just gets worse from there. So that is a real problem now. And that is where they think the next focus needs to be. And that is what I wanted to touch upon slightly here in this talk today. Next. Next. All right. Yeah, so quickly, how cancer cells develop drug resistance. So one thing is, there are many different ways how this can happen, right? So one thing is, when the cancer develops initially itself, right, they think that it's not necessarily just one type that's within the tumor, but there are many different types of cancer. And therefore, when you give one drug to the cancer, that that those cells that respond to it die off, but then the cells that do not respond to the drug just stay there quietly, and then after a long time it just keeps growing, and then after a long time, once it reaches a threshold, it just continues to grow. So that's one reason why it comes back and does not respond to drugs, right? So the second thing is tumor cell adaptation. So this is a very cool thing, and we did some basic science research in this area. So here, what happens is cancer cell, the tumor is very, very, very adaptive in the sense that, so let's say I mean, there is a tumor, and then you're trying to attack it with a drug. You keep giving a drug, and then what happens is after a while, because the cancer now understands how this drug works, so it tries to find alternate ways of escaping the drug. So it creates mutations to get out of to not react to the drug. So mutations happen genetically because of the genetic lesions and everything. So those populations expand, and therefore those will keep growing, which will not respond to drug, and that's one mechanism. The third mechanism is instead of the tumor actually actively participating, it's the microenvironment, the outside of the tumor, that is working to give it the resistance. So yeah, that's another part I don't want to talk about today. The fourth one is the therapy failure. This is because very simple reasons, like the tumor is in a very unreachable place, and therefore you're not able to give the drug into the tumor. So it does not, no matter how much drug you give, there are some barriers. So the drug is not able to reach the tumor. So that also happens, and therefore it just keeps growing and evades drug. So okay, so you moved on to the next one already. Okay, so this one, right? So this I'm just trying to show what happens at the molecular level, right? So what happens is, so if you look at the left panel, so you're seeing that this is what normally happens, right? Say, for example, you have a receptor, the white thing, it's called a receptor. Let's say that is what is going crazy, and that's why there's cancer. So that is the pathway. So that white thing activates something, the RAS and the RAF and the MEK and the ERK. So that's the pathway that's going crazy, and that's why the cancer happens. But so how you tackle that is give a drug that will bind to the Y receptor, and therefore the pathway is blocked. So that is how the drug is working, right? Now, what happens is the cancer sneakily will do modifications to this pathway. So if you look at the right side, so what the cancer cell could do is it could create a mutation at the point or somewhere so that the drug cannot bind to it, and therefore the drug is, even despite the fact that you give the drug to the patient, the drug is not able to bind to the receptor, and therefore the receptor is active, and therefore the pathway continues, and therefore nothing happens in the patient body. I mean, the drug does not do anything to the patient. So that is one way. Another way is, despite the fact that the Y receptor is bound to the drug, the cancer cell just figures out a different way of activating the same RAS, RAF, MEK, ERK pathway, and therefore it's able to continue the pathway without the Y receptor, and therefore it just continues being cancerous. Another mechanism is, despite the fact that you give a drug and the Y receptor is bound to it, there is some other mutations downstream in the pathway. Say, for example, in the RAF protein, which makes it active despite everything else. So it just remains active no matter what, and therefore that is one way. So the other way is, of course, it just activates some other bypass mechanism. So again, what I'm trying to say here is that what happens at the molecular level, what happens when you give a drug, and how the cancer sneakily tries to gets a way of finding some other mechanisms to progress. Next. Okay, so knowing all this, how do we strategize? One way is early detection, and that's exactly why there's a lot of research going on here. They're trying to understand, and that's why screening is also important. So that's why people are pressing to get cancer screening done, no matter what. Especially if you have a cancer running in the family, get cancer screening done. So you can catch cancers or tumors in your body very early, and therefore you can work on it very, very early, and that gives you a much better chance of killing it off. So that's one way. The second way is deeper response. This is you are trying to, when you start the treatment, give the most you can drug-wise to the patient. So a lot of damage is done to the tumor at the start. So that's what I mean by upfront administration of more potent inhibitors. So give everything in the beginning, and then let most of the damage be done to the tumor, and then we'll take care of the rest later. That's one way of doing it. That's a very aggressive way of attacking cancer. The third way is keep monitoring. Just keep seeing how the tumor is growing, how the tumor is changing or mutating in response to drugs. Like I said, because you give drug A and the cancer finds a way of mutating and not responding to drug A, it does something else. And so you have to keep monitoring the patient to understand, and try a different drug after a few days, so the cancer does not have a way of evading all those different drugs. So that's one reason why clinicians try combination therapy these days, or they follow one drug with another drug. They don't continue the same thing again, just to confuse, basically, the cancer. Next. So lastly, I just wanted to quickly touch on this. So this is, again, a new area getting a lot of attention. So this is another way of overcoming resistance, right? So what happens is, in a cancer cell, when you give a drug to a cancer cell, what the cancer cell does is it has all these drug pumps on it. So when you give a drug, the drug goes into the cell, but the pumps on the cancer cells will just throw it out of the cancer cell. So what happens is, so effectively you are giving the drug, it goes in, but it gets kicked out. So nothing happens. The drug is not able to act in the cancer cell, and therefore the cancer cell lives and survives. So one way to tackle this is use drug pump inhibitors. So you are stopping the drug pump from acting. So the drug pump becomes inactive. Therefore, the drug goes in, but does not get out. So that it stays in and does what it needs to do. And again, there's a lot of research going here. So and a lot of combination research going here. Like there is a lot of different drugs, drug pumps that work in different kinds of cancer cells. So what they're trying to do is combine all these different inhibitors and try to see what works the best. Next. So yes, I think we have come to the final slide. So this is all I had to talk to you about today. So quickly to summarize, what we spoke about was two very hard ways of tackling cancer. One is hacking the immune system. We talked about inserting an antibody to make the immune system recognize the cancer cell. We talked about the PD-L1 and PD-1 inhibitors, the immune checkpoint inhibitors. We talked about the cancer vaccines, which is where I talked to you also about the computational ways in which some really cool things are happening recently, and the CAR-T. So these are the immune therapy ways. So the second one I talked about how to overcome chemoresistance, and I just touched on two things. One is early detection, biomarkers to detect cancer early, and then deeper response, attack strong at the beginning. And the second one is drug pump inhibitors to stop the drug pumps from expelling the drugs outside the cancer cells. So yes, next slide, and that's my thank you slide. That's all I have for you.
Thank you, ma'am. Thanks for presentation. Madam, can you elaborate the positive side of immunotherapy, madam, in the cancer research?
Post- uh, yes. Sure. So positives, I would say the main thing is you're trying to hack your own system. It's nothing. So let me go back one step and try to explain why it is better than chemotherapy. Of course, they work in tandem. They work parallelly. So you have to give chemotherapy, of course, and then of course do radiation, of course. So everything works together. But then the plus side of having immunotherapy is that you when it is when you are giving a lot of drugs into the system, there also needs to be a way of continuing the attack, and that's why immunotherapy is good. So it works well with chemotherapy. um, yeah, that's the main thing I can think of.
Okay. Thank you, madam. What is the economical side of the immunotherapy, madam? Whether it is cost effective or cost effective?
Oh, that I'm
Patient.
not sure about. I don't know yet, because like most of the things that I talked about is mostly in the clinical research phase, right? And mostly tried in very advanced cancer patients in their advanced level of cancer, right? So it's not, a lot of them are not treatment standard of care stuff. So I'm not exactly sure about the economics of it yet. It's too early to say that.
Thank you, madam. Any other, yeah, there is a question in the chat box, madam.
Okay, let's see.
Can you explain about the cancer vaccine? You explain about cancer vaccine in your own slide, but
Uh-huh.
the vaccines are only used to skip the spreadable disease. The cancer is back to the human body in genetic and environmental reason only.
You're asking if it's only because of mutation?
Yes, madam. So the question is,
Uh-huh.
it is not spread on. It is not a communicable disease. Why
Right.
there is need of vaccination?
Oh, no, no, no. Okay, right. No, so the reason why it's called vaccination, okay, it's not because it spreads from one person to another. That's not the reason why it's called vaccination, right? Yeah, the term, I think it's probably misleading, but then the reason why it's called vaccine is because you're trying to prevent something from doing much damage before it actually hits you, right? So that's why it's called cancer vaccines. So these cancer vaccines are used, say, for example, let's say, for example, you have, somebody has, okay, somebody has in their family, it runs the cancer runs in the family. Say, for example, breast cancer runs in the family. There's HER2 positive, something like that runs in their family, right? So when that happens, you know that your mom had cancer, your grandmom has cancer. So you think that you have a high chance of getting cancer. So what you do is you would go and get yourself screened to see if you have the genetic lesion, if you are predisposed to cancer. So, and then let's say you do the screening and then you find that you uh, do have that lesion, you can develop cancer in the future. So in that case, what you do is go and get the cancer vaccine. So it's like a preemptive strike. So you get this done before it actually hits you. So this is one reason why it's called cancer vaccine, but it's used broadly, but then yeah, that's why it's called cancer vaccine. It's not because it's communicable. Yes, to answer your question, yes, it's mostly because of genetic lesion. It has to be something happening in the genes for the cancer to, because if something is wrong with just one cancer cell, nothing is going to happen. I mean, one cell, nothing is going to come out of it. Only if that is in the genes, it is going to be passed on to its progeny, and that's going to go crazy only if it's in the genes. Do you see what my point is?
Yes,
So,
madam.
yeah.
There is another question from the same person, Mr. Raghavendra. For leukemia patients, there is any other drugs to cure the cancer without blood transfusion.
Say again. I'm sorry. Which one is it?
The patient with blood cancer are treated by blood transfusion now.
Yes.
And there are any drugs which can be treat the patient.
Say again. Your last part got cut off.
So, can we cure blood cancer with any drugs? Because
So let's see.
I
Uh-huh.
thought he saw a lot of Tamil movies, older Tamil movies. The heroes are having leukemia. So he want to clarify those parts.
Yes. I'm trying to think what we are doing. So, yes. So, most of the immunotherapy that we talked about today, right? Most of them, they are working in leukemia. There are trials in leukemia. If you're really interested, I would recommend for you to go and look in clinicaltrials.gov, to see what's going on in leukemia research. Yes. To answer your question, yes. I'm not able to think of any one drug for some reason, but yeah, there's a lot going on.
Okay, madam. There is another question from Mr. Athanadi, sir. The way of identifying cancer in initial stages.
The way to identify, yes. So this is another region. This maybe I can give you some examples from my own research, right? So yeah, biomarker. That is one huge area where. Hello.
You can
Biomarker
proceed.
research is, yeah. So one. I'm sorry, my dog is here. All right. Um, so what? Yeah. So where was I? What was he saying? Um, oh, the cancer screening. Yes. So biomarker research. So what they're trying to do is, especially if you think you're susceptible to getting something in the future, you would go and so there are many tests already right now. You have many kinds of screening. You have screening from the blood also. If you anyway, so there are many different screening panels, and what they are trying to develop right now is screening. There are different. I'm trying to. I'm talking. I'm trying to think of so many different things now. But anyway, for example, okay, let me give you one example. So one way of identifying early is the circulating tumors DNA, the circulating tumor DNA, ctDNA. I don't know if you've heard about this. Um, so this is something that, so if you have tumor in you, right? So that will release some DNA. It's called ctDNA. It's circulating tumor. So, and then every kind of tumor, like say, for example, if I have pancreatic tumor, I'll have a different kind of ctDNA. If I have esophageal tumor, cancer, I will have different kind of ctDNA. So, so what they're trying to see is, even if you cannot detect by any other mechanism, like say, for example, if you do a CT on a person, you cannot detect because it is in a very early stage of development. The tumor is really, really small. But when you do a CT DNA analysis, you will be able to uh, detect if the person has even that small amount of cancer in him. So that is one area where we are trying to work on. We are working on, and we are trying to see how early we can do the detection. So I hope that answers your question.
Thank you, madam. There is a question from Mr. Harigaran. The drug
Mm-hmm.
resistance in cancer cells is only due to mutation.
Yes, we just spoke of that just a few minutes back. Yes, mostly yes. Something has to happen in the genes, or at least epigenetically. Something has to happen for at least more than one generation of a cell for it to become completely cancerous. So yes, genetically or epigenetic.
Okay, madam. Thank you, madam. There is another question from Mrs. Parasakti. So there are a lot of anti-cancer vaccines for uterine cancers.
Uh-huh.
Whether they are working like they, my gynec told if injecting vaccine to girls before marriage, they won't affect for the whole life. Is it true or not?
Yes, there are some kind of vaccines. It's again, it depends. Like, yeah, we cannot generalize anything. There are, say, for example, I'm guessing HPV vaccines. That is one. I'm thinking that's the example that the person who asked the question is referring to, right? So yes and no. It depends on the cancer, the type of cancer. Say,
Okay,
for
madam.
example, HPV vaccine that prevents a specific type of cancer that happens because of a certain virus.
Okay, madam. Thank you, madam. Uh, there is a question from Dr. Rajabandi.
Uh-huh.
Can you give some more idea regarding synthetic neoantigens, which is used in cancer vaccines?
Synthetic, oh, the
Neoantigens.
synthetic neoantigens? Yes, sure. Yes, we did a trial, and we were. I was very an integral. I was very. I was working very closely with the team who was doing it, so it was kind of cool to see the whole thing. But anyway, so yes, it is. So, so okay, what we were doing was we just took what a very common kind of what do you call it, antigen, a cancer specific antigen that is seen in certain type of breast cancer. So we took that and synthesized that. I think it was a very short peptide, 11 or 12 amino acid peptide. We synthesized that and then used that to create vaccines. So I'm sorry, I forget what the question was.
About synthetic neoantigens.
Right. So yeah, so that's it. So like I said, there are many ways of synthesizing it. How we synthesize was just combine the dendritic cells with a neoantigen and then just give it back into the human body. But then, like I said, there are many different ways of synthesizing it. You can either do it inside the body or outside the body in petri dishes.
And one final question from Dr. Fatima.
Uh-huh.
It is vaccine is prevent genetically transmitted cancer.
Vaccine prevent genetically. Yes. Yes.
Okay, madam. Okay. Thank you.
Thank you, madam. Thank you, madam.
Thank you. Thank you for bearing with me and my technical difficulties. I will log off the call now.
I think principal is speaking, madam.
Okay.
Sir, we can't hear your voice.
Um,
Sir, we can't get your voice.